So next we have one more virtual presentation from Danielle Miranda. And Danielle is Research Assistant Professor at the University of Notre Dame.
Hi everyone, sorry I was planning to be there, but things happened, I couldn't be in person. So I will be presenting part of a project that I've been working for the last 2 years. This is titled Patterns of PFAS Abundance and Composition in Lake Michigan Prey Fish. So this project was supported by the USGS and also by the Sea Grant, Illinois, Indiana. I think you already heard a lot about PFAS, but I'll just keep going with my presentation and say that those eyes were I also just heard from John, they are just synthetic compounds. They are man-made compounds characterized by those strong carbon-fluorine bond, and they can be different composition. I always say that they pertains to their group of the PFAS has more than 10,000 compounds. Now we are already counting for 14,000 compounds, and they can be divided in different subgroups, compose this family tree. And so as we know, they can be in different applications, different industrial and everyday products, as from non-stick cookware to AFFF. And why do we care? From the human health and from the wildlife perspective, we care because they were already related to different malformation in fish and also in other kinds of wildlife, and also to the development of cancer, dysfunctions of thyroid, and other issues related to the human and wildlife health. We also know that PFAS can be found in different environments, and they get together with the high toxicity, and they can also rapidly spread in water bodies, and they have the potential to bioaccumulate. So nowadays we are inputting PFAS to the environment, to the aquatic ecosystems by several types of waste, like industrial waste, agricultural waste, domestic waste, and depending on their composition, they can be more or less available in the water. They can accumulate more or less, and then they can be transported to oceans and also be partitioned to the atmosphere. But in this presentation, we're gonna focus on the bioaccumulation of those compounds in prey fish, so that's why I'm highlighting this here. So the main goal for this study was to look at 21 PFAS compounds and measure them in 6 species of prey fish. So in partnership with the USGS and with their monitoring program, they collected and gave us actually samples of those 6 fish species, and a total of 211 samples from 6 sampling points in the Lake Michigan. So we have here Michigan, Wisconsin, and also Illinois. Briefly talking about how we extracted the samples. We analyzed all the samples with the LCM SMS to look at 21 target PFAS from different lengths, having short and long chain acid PFAS. And then we analyzed 0.5 grams of freeze-dried and homogenized samples with internal standards. And the extraction was made with methanol, sonicated for 30 minutes and centrifuged. And the cleanup step was made with AmphCarb. So we saw this methodology in other papers, and we adapted for our needs. Everything that I'm gonna show here, it's been combined for a publication. It's in the initial stage, but I just want to mention that. So let's go to the results. From that 211 samples. We had to combine some fish species, some individuals from the same species according to the nitrogen and the carbon content. And then, because we didn't have enough mass to do the analysis, and it resulted in 6-7 samples. So the 6-7 samples were from those 6 species. And as you can see here, we had to separate the slime sculpin because the concentration was so high. And it's also important to mention that the samples were combined from 2020 and 2021. And here we have the PFAS concentration. I'm gonna see the list of compounds in the next slide. So from PFAS were observed in all samples, regardless of species. And from the 21 compounds, we observed 16. 17, but I'll talk about one of the compounds that we didn't report. And the concentrations were in between 2.3 nanograms to 203 nanograms per gram. So yeah, PFPEA was the compound that we excluded from the sample set because we found a interference in the main ion when we were looking to this compound. And I can talk more about that after or in the chat if you want, if you know why and how we figure out that this was interference and not a actual compound. And those short chains here and a long chain sulfonic acid was not found in any of the species. Here, sorry, I just put it here now. So here's the list of compounds that we found in all the species. And you can see that this blue color, like always in all samples, we just had this question before about why PFOS is the main it's always the main compound. And the presenter explained pretty well that this compound was used a lot. And we still have some precursors that are breaking down to this compound nowadays. And also, this compound has the higher potential for bioaccumulation compared to all the other PFAS presented here. And it's also interesting to see that we also have PFOA here, this green compound. PFOA is not supposed to bioaccumulate as much as the other, let's say sulfonic acids as well, but it's not also as long chain carboxylic acids. But it's bioaccumulating here in those organisms. And I separated here the list of the main carboxylates that were accumulating in those fish, and they are basically from here to here long chain compounds. So then I am trying now, in the next slides, I'm gonna show you we are trying to understand the distribution of PFAS in the Lake Michigan and trying to look to the different species. As this was a, let's say, opportunistic sampling, we couldn't get the same fish species for the 6 places, that we analyzed. But then I tried to compare the same species in different areas and see what you could get from that. So here we have Frankfort, that is in Michigan, with the highest concentrations compared to the other places. But we have to think that slimy sculpin and the slimy sculpin was only collected in this area, so this is not representative. We cannot compare just looking to the location and combining all species. So I tried to break down the species in different locations, as you can see here. So for this, I'm just gonna show for the different species and different locations from now on, and say where we saw difference between the locations or we didn't see. So here again, Frankfort is showing a high concentration, but actually it's not significant different from the other place. And then we have, when we changed to bloater, again, we didn't see significant difference. It didn't include St. Joseph because we only had one sample from there. But for the other place, we didn't see, again, a significant difference between the locations. But then when we changed to deepwater sculpin was collected in in Michigan and also in Wisconsin. And we can see with deepwater sculpin a difference between east and west in the Lake Michigan. I didn't have the same plot for slimy sculpin because, as I said, slimy sculpin was only analyzed in Frankfort and with the highest concentration for all the preyfish species. For around goby, when we only have samples from this area, Frankfort and Ludington, and we also didn't see any difference between the two locations. So yeah, this is what we have for those different locations. We are now trying to figure out how we can use this data together with the carbon and nitrogen to understand these patterns for the PFAS distribution in this environment. So as preliminary results, we have that PFAS was found in prey fish and all prey fish from Lake Michigan. And PFAS composition and concentration in Lake Michigan do not seem to be site specific, but species specific. We also saw, like in previous studies, that slimy sculpin and deepwater sculpin, they are accumulating higher concentrations of PFAS than when compared to all the other species, to all other analyzed species, even when we compare to salmonids as well. And again, PFOS was the dominant compound in fish from Lake Michigan, even after 20 years that this compound was phased out. And as I said, this is a very persistent compound, but we still have other compounds that are breaking down and transforming in PFOS in the environment. Very quickly, I want to thank for everyone that assisted in this project. Mainly to the Sea Grant and the USGS and the Indiana Water Research Center that supported our project. Thank you so much.
Thank you, Danielle. We do have time for a couple of questions.
Were the specific species that you used, were the only ones that you were able to catch, or did you target those specific species?
Yeah, as this was a partnership with the USGS and they were doing the monitoring program, so we got the species that we are able to get, that they were able to donate to us.
Any questions? We have one more and then that will probably be our last question.
Did you measure the total organic fluorine at all, as far as kind of like a way to kind of close the total PFAS estimate?
Yeah, we didn't do that for this specific project, this specific part of the project.
Okay, thank you very much, Danielle.
Thank you.